trust that's okay? Okay. Okay, so today I'm going to finish up, hopefully, talking about membranes. And we got a good start on things last time. So when I finished, I had talked about that fluidity issue with membranes and how organisms change the composition of the fatty acids in their membrane lipids so as to adapt to the environment in which those cells are found. And hopefully that made some sense to you. This transition that I talked about is basically what you see on the screen here, going from a more solid-like state to a more liquid-type state. And that is facilitated by heat. So the warmer it is, the more you can tolerate the saturated, and the colder it is, the more you're going to want the unsaturated to make them be fluid at a lower temperature. Okay. Butter versus margarine, big debate out there. And the debate is moving, I think, very quickly. I should say quickly, but it's moving back in the direction of butter. It used to be that people always considered that margarine was so much better for you, blah, blah. Well, margarine turns out to be produced by the, partly by the partial hydrogenation of vegetable oil. So what happens when you hydrogenate vegetable oil? First of all, I didn't give you the definition yesterday, the difference between an oil and a fat. But the difference between an oil and a fat is very simple. They both have the same chemical composition. They're both triacylglycerols. But an oil has more unsaturation associated with the fatty acids within it than does the fat. So the oil will be a liquid at room temperature. A fat will be a solid at room temperature. And that's really the only distinction between the two. So you would predict, based on what you know about the structure of fatty acids and fats, that they would have more unsaturated fatty acids that would be in an oil than in a fat. Well, if you take an oil that's liquid at room temperature and you want to make something solid out of it, like let's say margarine or something like that, you have to change the chemical nature of the fatty acids comprising it. And so what companies have done for many years has been to do what's called partial hydrogenation of those unsaturated fatty acids that are found in oils, typically vegetable oils. What partial hydrogenation does is it adds hydrogens across those double bonds. And when it does that, it creates a more saturated fat. And of course, saturated fats have higher melting temperatures, so it's more likely after you hydrogenate that you're going to make something that's solid at room temperature, which of course margarine is. Now, the downside of that is that partial hydrogenation results in some chemical byproducts that cause problems. And those chemical byproducts include what we refer to as trans fats. And what these are fats that contain fatty acids that instead of having cis double bonds, I told you yesterday that almost all fatty acids produced biologically have cis bonds in them if they're unsaturated. If you partially hydrogenate those, when you have a polyunsaturated fatty acid, at a small percentage of the time, it will make a trans bond as a part of the chemical rearrangement that occurs. So trans fats are fats that contain fatty acids with trans double bonds in them. And there do appear to be some significant health problems associated with trans fats. Okay? A lot of them relate to cardiovascular disease, inflammation, and a variety of problems. You see many foods, and you guys are many of you are nutrition majors. You know what I'm going to probably tell you, but I'll tell you anyway. And that is, you see many foods that say trans fat free. Does that mean they don't have trans fat in them? No, it doesn't. It means that they have less than the FDA allowable amount, which is 0.5 grams per serving. So they could have 0.49 grams per serving, which is a pretty good chunk of trans fat that's in there, and still claim that they are trans fat free. All right, so be aware of that. If you see the words partially hydrogenated within a product description, that product has trans fat in it. Okay. That product has trans fat in it. Okay. This doesn't list trans fat. It lists saturated, monounsaturated, polyunsaturated. And you can see some very big differences within them. And the view on saturated fats is changing. It used to be absolutely, people said, oh, avoid saturated fats as much as you can. And now there are people who say, well, maybe we should rethink this a little bit, and I'm not going to wade into that. When I've waded into it in the past, people who watch my internet videos go, oh, no, 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 no. Oh, yes, 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 yes. Oh, no, 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 no. So I just go, okay, I'm just going to shut up about it until we decide what the story is on these. Okay, so I'm not going to wade into that controversy. Okay. Getting back to membranes, which is really what we're after here with this set of lectures yesterday and today. Membranes, as I told you, play very important roles for cells. One of the most important roles that it plays is that of a barrier, as I mentioned before. And that barrier is very, very effective. Okay? Most chemical compounds do not easily move across the lipid bilayer. There are only 4 common things that we'll talk about in here that move across the lipid bilayer without some facilitation. Those 4 things turn out to be very important and very simple. One of those, oxygen. Oxygen can cross the lipid bilayer without too much problem. Doesn't need any help. Second, carbon dioxide. Carbon dioxide is a product of respiration, so getting it out of a cell in a simple fashion is useful. The lipid bilayer doesn't pose much of a barrier to carbon dioxide. Third, carbon monoxide. Carbon monoxide has a shape very much like oxygen, and it slides through the lipid bilayer pretty readily, doesn't need any assistance in doing that. The last one of these is important. They're all important, but the last one of these is important and it's interesting. And it's the molecule water. Water. Okay? Water moves across the lipid bilayer fairly easily. Well, if we think about this, when you did freshman biology or you did freshman chemistry, you probably did some experiments with dialysis tubing. You guys do experiments with dialysis tubing? What do you do with dialysis tubing? You put some things in the dialysis tube that can't move across the boundary, and you put it into a solution, and what happens? Well, the tubing starts to swell, and it starts swelling because the water can move across the boundary to try to dilute out the thing that's in it, but it can't dilute it out because the thing inside of it is stuck there. So the concentration of the thing inside the tube is always going to be higher than the concentration of the solution outside the tube because the thing can't cross the barrier. So the tube swells and swells and swells because water keeps coming into it. Well, if you think about it, that's actually a problem that a cell has as well. You never thought about that before, but it's true. The cell has a similar problem of dealing with osmotic pressure because water can cross the lipid bilayer quite readily, but many of the things and most of the things inside the cell can't cross the layer without assistance. So cells are fighting a continual battle to stay alive and not burst. Because if you let that go forever and you don't do something about it, the water will burst the cell. Okay? Well, I'm going to talk later in the lecture today about some transport systems that the cell uses to manipulate the ion concentration so that it doesn't burst. Okay? Some transport systems that manipulate ion concentration so that the cell doesn't burst. The cell has to burn a fair amount of energy in order to keep from bursting. And that's true of every cell in your body and virtually every cell that lives in an aqueous environment. Well, that leads me to the topic of what you see on the screen here, which are membrane proteins. Okay? Things don't cross the lipid bilayer by themselves, need assistance in getting across the lipid bilayer. And one of the ways in which they do that is with proteins that are embedded in the lipid bilayer. So I'll give you a really good example. Your blood cells are loaded with a set of proteins called GLUTs, G-L-U-T. A GLUT stands for a glucose transporter. What a glucose transporter allows is for glucose to enter a cell. It will allow only glucose to enter, and it will pass through the protein that allows that to happen. We can think of this protein as being something like a gatekeeper, checking to see that it's glucose, letting glucose pass through, and letting nothing else pass through. Okay? There are two ways in which that protein can do that. One is through what we call active transport, and one is through passive transport. Active transport requires external energy. And I'm going to give you a definition here that students will frequently trip up on, so please pay attention. And that is this. Active transport will always, there's an always for you, it will always involve the movement of a molecule from a low concentration to a higher concentration. Active transport will always involve movement of at least one molecule from an area of lower concentration to an area of higher concentration, and that's why the energy is needed. It's like pumping water uphill. Okay? You're moving against a concentration gradient, and that takes energy to defeat. I said there were two ways for this transport to work. One was active transport, which I just described to you. The other is passive transport. Passive transport is also called facilitated diffusion. And you'll notice in the name facilitated diffusion that the word diffusion is the driving force that makes that happen. Diffusion always works from high concentration to low. So if I had, let's say I'm looking at a blood cell on the screen, and the outside of the blood cell is on top, and the inside of the blood cell is on the bottom, and I have a glucose transporter for protein number 1 right here. So long as the glucose concentration is higher on the outside than it is on the inside, which is true for most blood cells, then energy is not needed to move that glucose in. It will move in through the process of diffusion. Therefore, that would be a system of facilitated diffusion or passive transport. Same thing. Okay? Okay. Now, the reason I have this slide on the screen is to introduce 4 terms to you and introduce 4 different kinds of proteins that we see associated with membranes. Okay? The first of these proteins, labeled number 1, is called an integral membrane protein. You can identify an integral membrane protein by virtue of the fact that it goes completely through both layers of the lipid bilayer. Number 2, we can see, is completely embedded in the lipid bilayer, and we call it an embedded protein. Embedded proteins we won't talk about here, and they're not very common. Protein number 3, you see, is not linked to the membrane protein anyway, but you see it's associated with one of the layers. And for that reason, we call it an associated membrane protein. It may be interacting with other proteins on the surface. It might even be interacting with that polar component of the lipid bilayer, but it's not in any way stuck into the membrane. The fourth one here is what we call a peripheral membrane protein. And by the way, some books differ a little bit. Some books call 3 a peripheral. I call it an associated. All right? So in my definition, a peripheral membrane protein is a protein that is stuck in one part of the lipid bilayer, but not both. There are many peripheral proteins, peripheral membrane proteins. Now, there's one other that's not shown on this figure, and it's something I call an anchored membrane protein. It's actually fairly easy to understand. Let's say we took protein number 3, and we stuck a long fatty acid on it, and we stuck that fatty acid down inside of here. That fatty acid would serve as an anchor to hold that protein into the membrane. So an anchored membrane protein is the 5th kind of membrane protein. Okay. What was its job? Is that what you said? So they all have different jobs. So they may have many, many different functions. Anchored membrane proteins actually play some roles in the process of signaling that we actually don't get to talk too much about here, but if you're curious, I'd be happy to tell you more later, okay? But there's many, many roles associated with all of these different proteins. The most common role associated with number 1 is actually transport. And so protein number 1, the integral membrane protein, is one we'll talk about the most here. Here's the anchored, all right? You can see a good example of the anchored right there. There's a fatty acid sticking its tail in there. It's attached to a protein down here, and that anchor serves to hold it inside of the membrane. What would you call this guy right here? It's a trick question. It's actually got 2 things going for it. So it's basically an integral protein, but you can see it's also anchored. The cell doesn't want that one going anywhere, right? That protein is stuck inside that membrane. Okay. Now, I'm just going to show you this, and there's not really any big take-home message with this. But there's a model that has been proposed, and it's pretty accurate, to describe the sort of chemical nature of a membrane. And basically, this model says that a membrane is fluid. That's what we talked about yesterday. It's not a solid structure, it's a fluid structure, and the fluidity is important to its function. We know that things in membranes can move to varying extents. They move to varying extents. The ones that move the least are those that are the integral membrane proteins. They tend to be less mobile than the other proteins are, but virtually everything inside of a membrane can move because the membrane is fluid. If we look at this figure, there is one thing I'll show to you that's kind of interesting, and that is you see various things called oligosaccharides, and you see various others of different colors that are out there. These are all carbohydrates, sugar-type molecules that are linked up here in chains. And you'll see that they have a variety of shapes and colors, indicating that they have different sugars and different numbers of them that are linked together. These turn out to be, in many cases, interesting because what in many cases they function as is to serve as identity markers for cells. And you'll see that if I were to show you a figure like this and I said, what's the inside and what's the outside, I hope you would tell me that up is the outside because we only see carbohydrates on the outside of a membrane. We never see them on the inside. Okay? That makes sense if we think about the carbohydrate being an identity marker. Because what it's saying is, hey, I'm a liver cell. Hey, I'm Kevin Ahern's heart cell. And you'll notice I said Kevin Ahern's heart cell because they also play roles in rejection of organs. When you hear about an organ being rejected in transplant, what's happened is that an organ got transplanted into somebody that didn't have the right markers or have the same markers as the person, okay, who the person who donated it didn't have the same markers as the person who got it. And so the immune system sees it as foreign because it says, that's not Kevin Ahern's heart. Okay? Blood cells. When we talk about blood types, blood types are determined by carbohydrates on the surface of their membrane. Okay. Well, let's talk about membrane transport. I've already started talking about the first part, which was facilitated diffusion, or what this figure here called passive diffusion, or passive movement, passive transport. Basically, it says that things will move, if they can, from a high concentration to a low, and it doesn't take any energy in order for that to occur. This figure, which has an equation on it, which we're not going to calculate, don't worry about that. Okay? This figure with the energy equation is simply there to tell you the amount of energy associated with that difference in concentration. The higher the concentration is compared to something else, the more force will be present to move from that higher concentration to that lower concentration. You know this inherently. If you take a glass of water and you put a drop of ink into it, when it first goes in there, it's a little drop of ink, and you start seeing it spreading out with time, and it's spreading out with time because it's moving from an area of high concentration where the ink starts out from. You come back a half an hour later, and the ink has diffused all the way through the rest of the glass, and it's gone from a high concentration out to an even and lower concentration. That's a tendency. If we want to go from a low to a high, we've got to expend energy, okay? And that requires active transport. Now, active transport, I'm going to remind you, always involves the movement of at least one molecule from an area of low concentration to an area of higher concentration. Always at least one. Okay? We may see some things going from higher to lower, but if we see at least one going from lower to higher, then we know we've got active transport. Active transport uses a variety of energy sources. One of the most common mistakes students make on my exam is they will try to tell me that active transport requires ATP. It does not. Some types of transport involve some types of active transport involve no ATP whatsoever. Okay? Well, here's one that does require ATP, and it's a transport system that is abundantly found in cells. And it's abundantly found in cells because this transport system plays that role I told you about earlier of messing with ion concentration so as to keep water from diffusing in and exploding the cell. This transport system is used for a couple of things, one of which is what I just told you. It's called the sodium-potassium ATPase system. I'll repeat that. The sodium-potassium ATPase system. Now, there's a bunch of steps on the screen, and we're not going to go through all of the steps. But I'm going to tell you the highlights of what's happening here. Okay? In this system, I told you that the cell is messing with ion concentrations, and based on the name of what I just told you, you should guess that the ion concentrations being messed with are sodium and potassium, and that's correct. What this pumping system does is it takes sodium ions, 3 of them, and you'll see them right here, from the inside of the cell, and it pumps them outside of the cell. Okay? It takes sodium ions from inside the cell, and it pumps them outside the cell. When I say pump, I'm generally meaning against a concentration gradient. And that's exactly what's happening here. Before we start, there's a higher concentration of sodium outside the cell than there is inside of the cell. To move them from inside to outside, I'm going against a concentration gradient. I'm going from low to high, which means I've got to expend energy, and the energy is being expended in the form of ATP. Okay? That's the first part of the system. The second part of the system is the potassium part. So in the first part of the system, 3 sodium ions get kicked out of the cell. In the second part of the system, 2 potassium ions get kicked into the cell. And that also is against a concentration gradient. Okay? That's also against the concentration gradient. So what's happening is we're creating an artificially diverse set of gradients. Higher sodium out, higher potassium in, and the cell balances this in a way that water does not diffuse into the cell. Now, it's more complicated than we're going to talk about here, but that's basically what's happening with this. If you shut this system down, you would expect that the cell would, first of all, probably burst. Okay? And then the concentration gradient would even itself out. Now, this concentration gradient, high sodium out, high potassium in, the cell uses for a variety of things. It can use that high sodium concentration to actually carry other things into the cell because sodium wants to come in. High out means it wants to come in. There's a pressure for it to come in. One of the places where that sodium comes rushing in is in your nerve cells. Your nerve cells use these two gradients to transmit nerve signals. When you burn your finger, what's happening is there are nerve cells that are opening up little gates. And those little gates are specific for sodium ions. If you take a cell and there's more sodium outside than inside, what's going to happen when you open up a gate? Sodium's going to come rushing in. That's the first part of transmitting a nerve signal. Okay? Now, I'm not going to go through nerve transmission, but that's one place where that gradient is used to do something. And the beauty of that gradient is when you have something at a high concentration here and a low concentration here, and you open up a hole between the two, that gradient moves really, really fast. The ions move in very quickly. And that's important because if you want to transmit a signal to, hey, stupid, you put your finger in the fire, get it out of there. Oh, you want that information to go to your brain pretty quickly. And that's what's happening in that concentration gradient. You open the gates up, things come in quickly, and all of a sudden the brain goes, whoa, he's got his finger in the fire. Questions? Clear as mud? Everybody's looking tired, like, when's he going to hand out those exams? He will in time. Okay, notice I did not go through the steps here. I told you the overall picture of what happened. Okay, so I spared you that. Here's an example of a place where ATP is not used to do active transport. Okay? It's a place where ATP is not used to do active transport. It's a pump that bacteria use to move lactose in. Okay? Lactose is a sugar. And bacteria are always looking for energy sources. And lactose, because it's a sugar, is an energy source. Lactose, like most molecules, does not move across the lipid bilayer by itself easily. It needs assistance getting in, and the assistance it needs in getting in actually comes from this lactose transport protein. It's called lactose permease. How does it work? Well, to tell you how it works, we first have to understand what's happening over here on the right. What's happening on the right is that this bacterium has something called a proton pump. Proton pumps are quite common in biology. Your cells are full of proton pumps. And not surprisingly, what a proton pump does is it pumps protons. It moves them against a concentration gradient, taking them from low concentration to high concentration. The impetus for that pump could be a variety of things. I shouldn't say impetus, but the energy source for that pump could be a variety of things. It could be ATP. In the case of photosynthetic bacteria, that energy source is light. That energy source is light. The energy source as light is a pretty cool one. I'm going to tell you about it because later in the term I'm going to tell you how to make a photosynthetic fish, and it relies on this. Okay? Let's understand how this pump works, and then we'll understand the rest of this. Okay? A pump that responds to light does the following things. It's a protein. It's an integral membrane protein. And in the middle of this integral membrane protein, it has a single molecule of vitamin A. Vitamin A. Okay? Vitamin A is important for vision. You know vitamin A is important for vision. And it's important for vision because vitamin A is light sensitive. Vitamin A is full of double bonds. And when vitamin A is exposed to light, those double bonds will shift from trans to cis. And that's what actually helps us to see. Because our nerve cells in our eyes are sensitive to that trans to cis transition. Well, bacteria use it for a completely different purpose, and it's really cool. The purpose that they use is as follows. Imagine that I have a fatty acid. That's kind of what the end of vitamin A looks like. It's a polyunsaturated fatty acid. And it starts out with all of its bonds in the trans configuration. My arm, or my leg, perhaps even better, my leg, when it's straightened out like this, is in the trans configuration. Okay? All the bonds are trans. When light hits the vitamin A that's in my leg, what happens is it moves to a cis configuration. And cis bonds, you know, involve bends. My leg is just bent if you're watching on camera. Okay? When it gets hit with light again, it goes back to trans. Cis, trans. And I'm showing kicking with my leg here because that's exactly what's happening with the vitamin A in here. It's kicking protons out of the bacterial cell, out of the cell. It's going against the concentration gradient. It's creating a concentration gradient. There's more protons out than there is in. And it's happening because of that kicking that happens in the presence of light. Everybody got that pictured? Well, once I've created a concentration gradient, those protons are greater concentration out than in. And by the way, protons don't move across the lipid bilayer by themselves either. I gave you the 4 molecules that do, and protons were not one of those 4 molecules. Protons, if you give them the opportunity to come in, they will do so, very much like the sodium ions. If I gave them the opportunity to come in, will do so. The difference here being they don't come in by themselves. They only come in when they carry lactose. They only come in when they carry lactose. So the concentration gradient of the protons are moving from high concentration to low, and they're carrying with it lactose that's going from a low concentration to high. This process is an active transport system because lactose is moving from low to high, even though protons are moving from high to low. We're using the gradient of the protons to drive this process. Okay. There's one other way, and we're getting near the end here, relax. So I know I'm going a little fast, but I want to get through this before I hand out the exams. Okay? There's one other common way that things get into cells besides what I've told you. So first of all, I said 4 molecules can move across the lipid bilayer by themselves pretty readily. That's one way. Second way, I said they can move through special chambers or special transport systems, like integral membrane proteins, to get into a cell or get out of a cell. We can get these out of a cell as well as in. They go both directions. Okay? A third way actually relates to cholesterol. Okay? Cholesterol, we'll talk about later, is a really odd molecule. There's an awful lot we don't know about cholesterol. But our body needs cholesterol for several things. One of the things I told you yesterday was we need cholesterol for membranes. I'm going to remind you of something I said yesterday. When I said yesterday that cholesterol was necessary for extending the fluidity of membranes, it means that that temperature range over which their fluid can be extended with cholesterol. Cholesterol doesn't raise or lower. It simply widens that range of fluidity for a membrane. Okay? Well, that means that cholesterol is important for cells to have. And it turns out that cells have several ways of getting cholesterol. One, they can make it. 2, they've got stores that are present in the body that can be used. 3, they can get it from externally. And that's what you see on the screen. This mechanism shown on the screen has a name. Excuse me, it's kind of a long name. It's called receptor-mediated endocytosis. How about I put that in the highlights? Receptor-mediated endocytosis. And basically what that means is that the cholesterol is encased in a complex called an LDL. Everybody's heard of LDLs, am I correct? LDL stands for low-density lipoprotein, and it's just a way of moving lipids in the blood. The lipids can include fat, they can include cholesterol, and they can include fat-soluble vitamins. It's a way for the cell to get lipids inside of themselves. What happens is the LDL, which is floating around in our blood, attaches itself to a receptor on the cell surface, and then the cell engulfs that thing and basically eats away the middle. So this method is another way of getting things into cells. Receptor-mediated endocytosis. Yes?
This one specifically for cholesterol?
Good question. Is it, okay, her question was, is this specific just for cholesterol, or is it used for other purposes? It will work for anything that's in that LDL. As we'll talk in the class later, one of the most abundant components of an LDL is cholesterol, but it's not the only component that's there. So there are other things, including fats and fat-soluble vitamins, that can be internalized in this way as well. But cholesterol is pretty abundant in an LDL. All right, well, we've gotten through the heavy stuff. Let's go. I'm going to talk about fat-soluble vitamins, then I'm going to finish up with prostaglandins, and then I'll pass out the exams, okay? You guys know about fat-soluble vitamins. You know there's 2 kinds of vitamins, right? Water-soluble, fat-soluble. Fat-soluble vitamins we have to be a little bit more careful with than the water-soluble vitamins. We don't worry about getting too much vitamin C. Okay? You need vitamin C by the gram, by the ounce, and we don't get too much vitamin C. But the fat-soluble vitamins we have to be a little bit careful with, particularly vitamin A. Vitamin A is actually toxic in high doses. If you ever go to the Arctic and they try to feed you polar bear liver, please remember that polar bear liver contains toxic levels of vitamin A. So say, no, I don't want that polar bear liver if they offer it to you when you're up in Juneau, okay? Or whatever, okay. Vitamin A, all right. Functions of vitamin A include vision. Here's the isomerization of vitamin A, by the way. Okay, hold on. Here's the structure of vitamin A. I said it looked kind of like a fatty acid. That fatty acid side chain, there's all those trans double bonds. Those trans double bonds can isomerize in the presence of light. And look at this. We see it going from all trans to a cis, and the leg has just bent, kicked, kick when it is trans, bend when it is cis. That's happening in your eyes. The same thing is happening in your eyes. The same bond is happening in your eyes. Okay? Vitamin A is also called by a couple of names. Retinol is the alcohol form of vitamin A, the OL indicating alcohol. Retinal, the aldehyde form of vitamin A, and you can see the aldehyde here on the end. There's retinol, the alcohol. There's retinal, the aldehyde. Retinal is the form that you have in your eyes. Retinol is the form that gets stored in your liver. There's also an acid form. It's called retinoic acid. That is, if we take that aldehyde and we oxidize it to a carboxylic acid, we create retinoic acid. And retinoic acid plays roles in cellular differentiation. Plays roles in cellular differentiation. Okay. Vitamin D. Vitamin D is made from cholesterol. It is therefore a steroid. And vitamin D, I hope everybody knows, in our bodies is made as a result partly of action of sunlight, converting this form of 7-dehydrocholesterol into vitamin D3, which you see going on right here. And you can see the catalyst for that is ultraviolet light. It means it's why you want to get a little bit of sun. You don't want to get too much sun, but you want to get a little bit of sun. Okay? Vitamin D goes, there's the modified form of vitamin D3 here goes through several forms. I won't go through all those forms for you. But suffice it to say that vitamin D is important for the proper absorption of calcium and phosphate. It's important for strong bones. Vitamin D is a compound that Dr. Fritz Gombart works on in the Linus Pauling Institute. He's a member of my department. He's a colleague of mine. And he's also shown very important roles for vitamin D in a healthy immune system. In a healthy immune system. Vitamin E, another fat-soluble vitamin. One we don't have as much information on. There are no known deficiencies in humans. The consequences of that aren't completely clear, but vitamin E is thought to play some sort of a protective antioxidant role. Vitamin D is commonly found in membranes, and it may help protect membranes against oxidation. You can see, again, here's this long tail. You can imagine that long tail being stuck somewhere inside of a membrane. The last one's vitamin K, and the only thing I'll say about vitamin K is that vitamin K is important for blood clotting. Vitamin K is important for blood clotting. Okay, a very quick and dirty mention of prostaglandins, and then I'll finish, okay? Prostaglandins. Prostaglandins are these odd-looking compounds on the screen. Prostaglandins are made from arachidonic acid. That's one of the polyunsaturated fatty acids I talked about the other day. Arachidonic acid has 20 carbons. Okay? 20 carbons. Okay? It has multiple double bonds. You can see 4 double bonds right here. Something that has 20 carbons we call an eicosanoid. E-I-C-O-S-A-N-O-I-D. The conversion of arachidonic acid to prostaglandins is catalyzed by an enzyme that has a couple of names. I'll tell you what they do in a second, but first, how we make it. It's catalyzed by an enzyme that has a couple of names. One of the names is prostaglandin synthase, or PGH synthase. They call it the same thing. The other that you may more commonly hear is known as a cyclooxygenase. Same name, same enzyme. I shouldn't say same name, but same enzyme, not same name. Cyclooxygenase. You might have heard previously of things called COX inhibitors, COX. COX stands for cyclooxygenase. Now, I'm going to tell you what they do, and then the COX inhibitor thing will make a little bit more sense. All right? What prostaglandins do are a variety of things. They stimulate uterine contraction during birth. They're associated with things that cause inflammation. And prostaglandins are most commonly associated with pain. They cause localized pain. If you can inhibit the synthesis of prostaglandins, you have a painkiller. Aspirin inhibits cyclooxygenase. Aspirin is a painkiller. You take aspirin for a headache because it inhibits prostaglandin. I'm sorry, prostaglandin synthase, the cyclooxygenase. It inhibits the production of prostaglandins. Okay? There are other things that people have studied to inhibit cyclooxygenase. They include compounds like Vioxx and other things that aren't currently on the market because some of those were found to be nasty and cause other problems. Ibuprofen is good, doesn't cause a problem. Aspirin, good, doesn't cause a problem. And they're both in the category we call non-steroidal anti-inflammatory drugs, NSAID. That's what they're called. Couldn't think of the acronym. Okay. All right. Now, we could do a song. However, you guys, I know, want your exams, so I'm going to save the song until the beginning of the next class period. How's that? All right. So, please come up, and I'll get your exam as quickly as I can.